Dr. Haidar is an associate professor in the Department of Biomedical Engineering and Department of Medicine at McGill. He leads an engineering and a clinical team to develop dosing algorithms for insulin pens and pumps in type 1 diabetes. So one of the things he's done is that in his efforts to translate these research findings to clinical care, he has worked with a lot of different companies. Just to mention a few: Eli Lilly, Bigfoot, which is now called Abbott, Agametrics, Adosia, and Camp Karawanis. So he's also the program director for the Professional Masters in Translational Biomedical Engineering that trains engineers for careers in the biomedical industry. So thank you very much, Dr. Haidar. It's all yours.
Thank you for the invitation. So I'll give you an overview about the field of diabetes technology and also about the work we're doing here at McGill. So I'll just give some quick, very simplistic background on glucose physiology. So glucose levels are tightly controlled mainly by two hormones in healthy individuals, so insulin and glucagon. And insulin brings sugar level down, while the glucagon increases blood sugar levels. And if an individual eats a meal, then the pancreas secretes insulin to basically allow the body to use the carbs in the meal to generate energy. And if someone is doing exercise, then the glucagon level increases so that the body can use glucose level for exercise. So sugar levels are tightly controlled by these two hormones, mainly. There are other hormones that play part, but these are the two main hormones. Now, if someone has type 1 diabetes, then they lose the ability to secrete any insulin. And before 1922, type 1 diabetes basically was a death sentence. If you're diagnosed with type 1 diabetes, you're expected to die within a few weeks, maybe a few months. And what happened in 1922, Banting discovered insulin in Toronto, and basically that really transformed type 1 diabetes from a fatal disease into a chronic disease that requires lifelong insulin replacement therapy. So this is a picture of the first kid that was treated with insulin, and you can see a big difference within 3 months. So Banting won Nobel Prize for the discovery of insulin in 1922, and he actually remained up to this date the youngest Nobel Prize winner in the category of physiology and medicine. And since then, type 1 diabetes became basically a chronic disease. So currently, people with type 1 diabetes, they need to give themselves exogenous insulin all the time. So the majority of people, they use insulin injections, so using pens, and they give themselves two types of insulin. So one type of insulin we call rapid-acting insulin that works for around 5 hours, and they need to give that with every meal they eat. And they also give once a day another type of insulin that we call long-acting insulin that basically works for 24 hours or even the new ones 48 hours. Now, some people here in Quebec, around 1/3, they use what we call an insulin pump. So it's a small portable device that you wear outside the body, which has rapid-acting insulin and a small catheter that is connected under the skin. And this device gives people continuous amount of insulin, and that you program it into the pump. So you can say at midnight, I need 1 unit every hour. In the morning, I need 2 units every hour, and the pump will continuously give you insulin in the subcutaneous tissue. Now, up until recently, everyone with type 1 diabetes, they measure sugar level with a meter. So you probably have seen this. So every 4-5 hours, basically, they take the meter, they prick themselves in the finger, and they measure their sugar level. And based on the sugar level, they decide how much insulin to deliver themselves, either through injection or through the pump. Now, this is basically a typical sugar level of someone with type 1 diabetes. So the target range, what we call the target range where we would like people with type 1 diabetes to maintain the sugar level in, is somewhere between 4 and 10 millimole per liter. So this is where our goal is. But most people really struggle to maintain their sugar level within the range. So basically, this individual here, they had a breakfast, sugar level goes up. They don't know that their sugar levels are high because they don't measure sugar level up until lunch. and then, oh my goodness, I'm at 20. They give themselves a lot of insulin, but then they give themselves slightly more than they need. And by the afternoon, they go to low sugar level, so we call hypoglycemia. They eat dinner, they don't give enough insulin, they go up, and so on. So because the target range is narrow, most people struggle to maintain their sugar level within that narrow target range because it's not easy for them to give the exact amount of insulin that they need. So maybe at lunch here, you need 2.5 units. If you give yourself 4 units or 3 units, you'll end up having what we call low sugar level. So you overdose insulin. Now, if your sugar level are high most of the time, you develop basically what we call long-term micro and macrovascular complications. So after 15, 20, 25 years, people start to have complications, eye, kidney, heart, and so on, and different complications. Now, if you have low sugar level, which means that you give yourself more insulin than you need, you develop short-term complications. And these are basically difficulty speaking, confusion, and in actually severe cases, it can lead to seizure and it can lead to death. So there's something called death in bed syndrome. So people give themselves a lot of insulin before they sleep, and basically they die overnight because of low sugar level. So people with type 1 diabetes, they have to struggle between short-term complications, so hypoglycemia, and high level, which is basically hyperglycemia. And if someone had one severe hypoglycemia, so one time they lose consciousness because of low sugar level, they end up having what we call permissible hyperglycemia. So they don't give themselves enough insulin because, long-term complication will happen in 15 or 20 years. So with the previous therapies, so people measuring their sugar level multiple times a day, and by giving themselves insulin through a pump or through an injection, what we know is that around 20% of the people only achieve the glycemic targets, which mean on average, their sugar levels are adequate to prevent long-term complications. And we can have a measure for this because HbA1c, which is a blood test that gives you average sugar level of the previous 3 months. So any question about the background before I talk about, can we have a question now or should we wait till the end?
We can wait at the end, but they can put the questions in the chat. Please feel free to put your question in the chat. Thank you.
Okay. So what happened in the last 10 to 15 years, we started to have what we call continuous glucose monitoring systems. So these are devices that were developed by multiple companies that measure sugar level in a continuous manner and relatively a non-invasive manner. So it's minimally invasive devices. And that really transformed the whole field of diabetes technology, because previously you only look at sugar level 4 or 5 times a day. Now actually you have your sugar level every 5 or every 10 minutes, and that allows people to have a smarter dosing mechanisms, either through the pump or through the injections. So these are the main, three in the market. Waveform technology is only available in Europe, and these people basically, anyone with type 1 diabetes can go and acquire these systems. So one of the advances that happened because of the development of the glucose sensor is what we call automated insulin delivery systems. So these are commercially available. The first one came to the market in 2017, so it's a really recent devices. And the idea is very simple. Now, you measure your sugar level every 10 minutes. You send that measurement by Bluetooth to the pump, and the pump has an algorithm that automates insulin delivery. While previously you programmed the pump to give you a certain amount of insulin at a specific time, irrespective of your sugar level, now the pump has basically become smarter, and if your sugar levels are high, start to give you more insulin than normal, and if your sugar levels are low, it start to reduce insulin delivery. Now, some people call it closed loop systems, some people call it artificial pancreas systems, and there is one particular group, they call the system bionic pancreas. But these are all terminologies for the same device that basically automate insulin delivery based on the sugar level readings. And again, the idea is very simple. People have thought about it for 50 or 60 years, but the only reason we can implement them now is the availability of these continuous glucose monitoring systems that open the field of basically closed loop therapy. So this is just one of our studies here at McGill. So this patient came to do a 24-hour at the gland, and the red line here is the sugar level as measured by the glucose sensor. The black line is the sugar level as measured by the gold standard. So we have a machine called WiseEye that is basically gold standard for measuring sugar level. So this was based on plasma glucose. So we had a nurse that would take blood from the patient every 15 minutes and we measure it. And you can see, generally speaking, the sensor is actually pretty accurate. Now the target range is this dashed line. So try to keep sugar level between these lines. These are grams of carbohydrate of each meal. So the lunch was 62 grams of carbohydrate. The dinner was 79 grams of carbohydrate. These are insulin boluses. So with every meal, the pump was giving a bolus. Now what you can see here is that the blue line is basically the insulin infusion by the pump. So you can see in the afternoon, the sugar level was dropping and the pump suspended insulin to prevent sugar level from further dropping into low glycemia. And the same for the evening. Here, sugar levels start to go up around 2 in the morning, and the algorithm start to give insulin to prevent sugar level from going in the high glycemic range to bring it down. So while previously people will get the exact same amount of insulin, with these new technologies, we're actually able to automate insulin to become smarter based on the sugar level readings. So there have been multiple devices that were developed by different companies and different academic groups, and most of them now in the commercial available. And people did randomized control trials to see how much these devices improve glucose control compared to conventional pump therapy. So conventional pump therapy means someone wearing an insulin pump, wearing a glucose sensor, but then they decide themselves how much insulin to give themselves every 10 minutes. Well, they don't do it every 10 minutes, but they program the pump and they can adjust this from time to time compared to the system that automatically adjusts insulin. And what we found is that the improvement in the proportion of time for which glucose levels are in target range is around 10-11%. So 10% around 2.5 hours per day. So basically, these studies showed that if you automate insulin delivery based on the sugar level readings, the amount of time you spend in the target range is increased by around 2.5 hours per day compared to the previous therapies where the insulin is basically pre-programmed into the pump. I see a question here. Yeah, so currently the pump and the sensor are separate, so they're not combined together. Yeah. So one thing you might notice is that if you look at the proportion of time for which glucose level in target range for all these systems, they're actually far from 100%. So that tells you these devices improve glucose control compared to the previous treatments, but we're still far from getting into basically perfect glucose control. Because if you are at 70%, this means that 30% of your time outside of the range, which is around 6 hours per day. So why it is difficult? Because, especially when you use the term artificial pancreas, people get the impression that that's it, everything would work perfectly. So the main reason is slow insulin absorption. So this is a picture from one of our study participants in a diabetes camp overnight. And you can see between 10 o'clock and 2 in the morning, the sugar level were above the target range. So the target range is these two dashed lines, and sugar level is above target range. Now the yellow line here is what normally this particular patient will be getting at home. So this is how much they program their pump to give insulin. And the algorithm is doubling at this point, tripling the amount of insulin they're getting, but sugar level still remain above the target and only drops a few hours later. And the reason it drops a few hours later because the absorption of insulin is very slow. So even though you might be smart in terms of algorithms, you don't have full control of the sugar level because they give insulin now, it works in half an hour, the peak action is 1 hour, and the entire duration of action is around 4 or 5 hours. Now, the other thing, there is large variability between individuals in the absorption of insulin. So this data from adult and pediatric patients that came twice to clinic, and we measured what you call the time to peak of insulin absorption. Meaning that if you bring someone and you give them an insulin bolus, and you wait until you have the peak insulin concentration in the plasma, you want to see how is that different between individuals. And some people had the peak at 30 minutes. Some people had the peak at 120 minutes. And also within the same individual, there is some variability. So this individual had the peak at 30 minutes in one visit and had the peak at 80 minutes in the other visits. So it looks like there's some reproducibility within the same individual, but there's also large variability between individuals and how insulin is absorbed into the body through an insulin pump. So what is next? So in our lab, what we're trying to do currently, we're trying to improve the proportion of patients that achieve glycemic targets. So currently what the recommendations are, people should try to spend more than 70% of the time above their target range. However, in a randomized controlled trial, what we observed is that only 1/3 to 2/3 of participants can achieve this target. So some of the studies I showed you earlier, when the average is 70%, that means half the patients, they actually don't achieve 7%. Half the patients, they have time in range below 70%. So we want to improve on this. And the other thing is that all current systems, commercial systems, is actually hybrid. Hybrid mean at mealtime, they need to look at the meal, and they need to do what we call carbohydrate counting, not calories counting, carbohydrate counting. So they have education about carbohydrate counting. They enter that number into the pump, and the pump will give the amount of insulin proportional to the amount of carbs that they're eating. And this is why in the previous graph I showed you, you can see the arrow of the meal, but there are also the arrow of the insulin boluses. And the reason we need this, because if you don't give insulin at the time of the meal, the sugar absorption is so fast that your sugar level will be too high before basically the insulin start to work. So this is what we're trying to do in the lab. We're trying to improve glucose control, but also try to actually alleviate the need of carbohydrate counting. And for that one, we use either approaches by combining drugs with these systems, or basically combining new algorithms with this system. So I'll talk about that in the next few slides. So one of the questions we're trying to answer that can adjunctive therapies improve glucose control? So adjunctive therapies mean a drug in addition to the closed loop system, and we borrow drugs from type 2 diabetes. And can adjunctive therapy simplify the need of carbohydrate counting? And I don't need to convince you. If you call someone and you tell them for every single meat you eat, whether you're at a restaurant, at a party, at home, you need to know exactly how many carbs in that meal. There's a whole kind of literature that shows this can actually induce negative psychosocial aspect, can induce basically negative dietary patterns, and so on. So we look at 3 drugs, or present today 3 drugs: empagliflozin, pramlintide, and semaglutide, or actually Ozempic. Yeah. So SGLT2 inhibitor, these are a new class of drugs that was developed for type 2 diabetes that are anti-hyperglycemic, so they bring sugar level down, that basically help the people secrete sugar in the urine. And in type 2 diabetes, there's the main study called the EMPA-REG trial, where they showed that it actually reduced mortality by around 39% in people with type 2 diabetes within 4 or 5 years. So people were very excited about this drug. It's a pill every morning that you take it. And there are other studies that show that it can have renal benefits and actually cardiac benefits independent of diabetes. So when people started to study these drugs in type 1 diabetes, there have been multiple studies with multiple drugs that are all SGLT2 inhibitors. It brings HbA1c level down. So HbA1c is a drug, it's a blood test that gives you the average sugar level of the last 3 months. Bring body weight down. It brings the total amount of insulin that people need also down by around 10%. However, all studies increase the relative risk of what we call diabetes ketoacidosis. So diabetes ketoacidosis is actually a life-threatening condition where the body starts to use fat to generate energy. And after a while, when you have too many, well, through that process, you start actually to produce ketone, and after a while, that can actually induce diabetes ketoacidosis. And traditionally, it happens when people don't get insulin. So people don't give them some insulin. After a while, they start to develop diabetes ketoacidosis. But with these drugs, we start to give diabetes ketoacidosis despite people getting insulin, except with one pill, which is low dose of empagliflozin. So we decided to take this drug and try to test them in type 1 diabetes. So one of the questions we try to look at is 25 milligram empagliflozin improve glucose control on people who are using closed loop therapy. So we did a randomized control trial on 27 adults. Their average A1C 7.7, where we had 4 interventions. Each one of them was 4 weeks. So empagliflozin 25 milligram with a closed loop system. The same pill with basically what we call sensor augmented pump therapy, which is the conventional therapy. People use the pump, use the sensor, but then they manage their insulin. And then we had the same 2 interventions with placebo. And we built our system where the algorithm was residing on a phone and then was communicating with the Bluetooth with the glucose sensor, and the algorithm also was communicating with a pump using actually Bluetooth. And we ran the study, what actually was before most of the closed loop systems were commercially available. So we had to build our own system. Now, just small thing in the background, even though this sounds like a pure clinical trial, this actually device is a class 4 medical device. And we had to get Health Canada approval, and class 4 medical device is considered the highest risk medical devices. And our application to Health Canada were around 1,500 pages. So they have to do like risk analysis and all kinds of testing, not just for the algorithm itself, but the user interface and the communication between the sensor and the pump. So we need to get that approval and build the system here, which actually had to be labeled and manufactured by McGill before we were able to do that pure clinical trial. And this is actually the system that we had to get approved by Health Canada. So some high-level description of how the algorithm works. We use what we call model predictive control, which is a technique that was developed in the 80s, mainly for chemical engineering to control chemical processes. And it's mathematical optimization over what we call dynamical space. So just high-level description of that, if at a particular time you know your previous sugar level and you know your previous insulin delivery, what the algorithm does using mathematical equation, it can predict the future. So the algorithm says, well, if I give this amount of insulin, the blue line, dashed line, the sugar level is predicted to take this kind of profile. If I give this other insulin profile, my sugar level will look like the new profile. If I give the third potential insulin delivered profile, my sugar level prediction will look like this third sugar level, this third profile. And we look at all potential insulin deliveries, and then we pick the sugar level that matches a criteria. In our case, basically the maximum amount of sugar level within the target range, or even closer to the middle of the target range. So once we choose the best profile based on the mathematical model, we only apply the first insulin delivery from that future predicted profile, and then we wait. And in 10 minutes, when we have the new sugar level, we compare that to what we predicted 10 minutes ago. Now, almost never they are equal because the mathematical model we are using are not perfect. So we use the difference between what we predicted and what we measured to update these mathematical models using what we call Kalman filtering, which is an approach commonly used in GPS, for example. Yeah. And current commercial closed loop system, the majority they use model predictive control. Not all of them, but actually 3/4 of commercial systems, they use this kind of algorithms. So if you look at the clinical outcome of the data, when we compared sensor-augmented pump therapy with placebo versus closed loop with empagliflozin, the improvement in time and range was over 4 hours. So people spend more than 4 hours of their day on average within the target range as compared to the previous therapy. Now, we wanted to measure keto level because this drug can improve or can increase ketosis, so high keto level. With placebo, we had not much difference between basically closed loop therapy and sensor-augmented pump therapy. However, when we gave people the drug of empagliflozin, the fasting keto level, so we were asking the patient to measure their blood keto level every morning for 30 days. We actually had an increased keto level. So which means that these people with this new drug, they were at a higher risk of potential diabetes ketoacidosis, especially people with lower BMI. And there is some physiological literature that look at why people with low BMI have potential increase of DKA. So we wanted to do another study with the low dose empagliflozin and people who do not achieve glycemic targets despite using this automated insulin delivery system. So we did a study where we recruited people who were using a closed loop system, but their time in range is below 70%. And we randomized them over 2 weeks, either 2.5 milligram EMPA, which is the low dose, 5 milligram or placebo. And the proportion of target range or the time in target range improved from 59 to 71 to 70%. So you can see with the low dose empagliflozin, which potentially has low risk of diabetes ketoacidosis, we're able to get the benefits of improving glucose control. And again, for that particular study, we had to develop a system. We have to get Health Canada approval before we run the study. That is a pure clinical study to answer pure clinical question. And now we're planning to look at a study over 6 months to actually compare 2.5 milligram versus placebo, and people are using commercial closed-loop system. Now, one thing, there is now a Chinese company that developed a continuous ketone monitoring system. So we plan to do a study where we test the accuracy of this device and then use it as part of our system to either dose, to help us to do the right amount of insulin, or even to help people realize that they might go into diabetes ketoacidosis and basically take action before they get into there. So this is more where we're heading in that little field, yeah. Now, the other thing what we're doing is that we're trying to develop what we call a dual hormone closed-loop system. So instead of delivering only insulin, we deliver another hormone called pramlintide. Now, this is pramlintide, an analog of a hormone called amylin, which actually co-secreted with insulin in healthy individuals. So this is data in 6 healthy individuals where you can see both insulin and amylin levels. It's completely lost in type 1 diabetes, but you get insulin replacement therapy. You don't get amylin replacement therapy. And the main action is slows gastric emptying. So when you eat food, it actually slows the absorption of sugar in the food into circulation, suppresses the glucagon secretion. So this data here, in healthy individual, when they eat a meal, their glucagon level remain constant. However, in type 1 diabetes, they have an abnormal increase in glucagon level. So if you give people pramlintide at the time of the meal, you can see in this study in type 1 diabetes, they prevent the increase in glucagon level by having more flat postprandial or post-meal glucagon level. So we said, well, can we develop a system where the pump, instead of giving one insulin only, we give insulin and pramlintide? So we built a system where we control 2 pumps. Now, we do not have access to a dual chamber pump. There's a company in the US developing it, but for research purposes, we actually control 2 pumps. One we give insulin, one we give pramlintide, and we adjusted our dosing algorithm. And after developing the system, we said, let's do a clinical trial where we compare this system to conventional conventional insulin alone closed-loop system. So this shows you one of our inpatient trials. So you can see here, basal, instead of giving only insulin, we're giving insulin and we're giving pramlintide. And at mealtime, so lunch was 63 grams, and instead of giving only insulin, we give insulin and pramlintide. So the green line here is pramlintide. And you can see, again, this is just a sample patient, but postprandial sugar level or post-meal sugar level are more stable. So we don't have this increase in sugar level after the meal that we saw with the insulin alone system. So we just recently finished a study where it was 2 weeks per intervention, 15 adults, 15 pediatrics, where we compared insulin and pramlintide with what we call simple meal announcement. So at mealtime, instead of doing carbohydrate counting, we're telling them just press a button at the meal, and that's it. So forget about carbohydrate counting for 2 weeks. Now, the other one was FIASP and placebo with meal announcement. So FIASP with a type of insulin. And the third arm was FIASP and placebo with carbohydrate counting, which is our control arm, which is basically the best available closed-loop therapy. So again, here what we said with the new dual hormone system that we developed, we also want to simplify the need of carbohydrate counting. So people just press a button at mealtime and forget about the meal. So this is the sugar level over the 2 weeks period, the average one. So the blue line is basically the control arm. With placebo, you can see when people are not doing carbohydrate counting, their sugar level are going up on average. However, with pramlintide, their sugar actually dropped down on average. So with this dual hormone system, we improved the proportion of time in target range by 1 hour, but on top of that, we actually alleviated the need of carbohydrate counting. Now, we wanted to know whether the improvement or the alleviation of carbohydrate counting did help patients' quality of life. So what we did, we worked with psychologists where we did interviews for each participant at the end of each intervention. So these are some of the quotes and some of the themes that came up. So a common theme that came up is that people felt less anxiety and stress around meals. Because again, they don't have to do any carbohydrate counting. And this is a quote from one individual. It made it easier with not having to count and eating. And it made it like less complicated and more like, oh, I'm hungry, I can eat. I don't need to like count stuff, which was nice. So this was an adolescent participants, which before the study, for every single meal they eat, they need to look at it, say this is 50 gram or 55 gram or 65 gram, because they have this portion of rice and that portion of salad and this banana. And then they need to do all that math, do it in the pump, and then they can basically before they eat. Well, currently they don't have to do any of that. Another adult said, I found it more natural way to operate, but also not having to do that calculation, it still removed a weight that is so usual that we don't realize it made a big difference. An adolescent said it avoided having my parents tell me it's my fault because I'm counting the carbs wrong. Because if you overcount or undercount, you'll overdose or underdose, it can lead to hypoglycemia. But actually, even when you have the perfect amount of carbohydrate entered into the pump, you can still have hypoglycemia because of all kind of basically stress, exercise, infusion set, absorption, and so on. Now, the other theme people felt restoring freedom and normalcy. So that came from multiple interviews. So this adolescent said, if I had a meal and I thought it was too big and I gave myself insulin, I didn't have to finish it, so that gave me more freedom. So previously, let's say you're hungry and you go to a restaurant, and if you have type 1 diabetes, then you say, well, this is the 100-gram meal, I'm going to give myself 10 units. Now, when you give yourself 10 units, that's it. You really have to finish your meal. Otherwise, the amount of insulin you took will actually lead to low sugar levels. So with the current approach, people don't have to have that kind of thinking. They can start eating, and whenever they want to stop, they can stop. They don't have to eat whatever they delivered insulin for. Now, one interesting comment was more negative. This participant said, I felt like the meal announcement strategy offered me liberty that didn't necessarily benefit my health, because now they don't have to count carbohydrates, so they felt that now they have a bit more freedom to eat what they want, and they felt it can negatively impact their health. So this was actually quite interesting feedback that we received. Now, the other thing we wanted to do with the insulin pamlintide is to do what we call fully closed loop system, so you don't even have to announce the meal. And the idea is that after eating, so this patient here had 58 grams of carbohydrate. This is, again, another study we did here at McGill. And you can see when the sugar levels start to go up, the system automatically starts to give insulin boluses after the meal. So what happened is that the algorithm was modified. So we have what we call a meal detection algorithm that detects the increase in the sugar level, knows there is a meal, and we start giving insulin. So we did a trial where we compared the insulin pamlintide fully closed loop system versus the insulin alone with carbohydrate counting in 24 adults. These are some graphs, so you can see that the sugar level are somehow comparable. These are the insulin outcomes. And we're replacing the insulin boluses with post-meal insulin with the fully closed loop system. And the time in range, actually, with the fully closed loop system was slightly degraded. And instead of 78%, we actually went down to 74%. But again, we had a fully closed loop system where people don't have to do anything at mealtime. Now, our study, we use 2 pumps to give 2 insulins. But ideally, we want to have basically one drug that combines both hormones together. So there are companies that are actually working on a co-formulation that combine insulin pamlintide or amylin in one drug, so people can put that into the pump, and they have a new dosing algorithm, and that's it. They get improvement in glucose control. And we did actually a study for a company called Adosha here at McGill. So Adosha, we did for them first in human clinical trial in a co-formulation that combined both insulin and a pamlintide. So this was really nice because initially we were doing 2 different pumps, and based on our outcomes, they developed for us this co-formulation, and we did for them a first in human clinical trial. Now, the other study we're doing with semaglutide, maybe not many of you are aware, they know semaglutide, basically Ozempic, so you might have heard this drug in the news, the drug that reduces body weight. And we're currently running the first study in type 1 diabetes, where we're looking whether the addition of semaglutide, which is basically Ozempic, can improve glucose control in type 1 diabetes. So people have done studies in obesity, in type 2 diabetes, but there hasn't been any studies in type 1 diabetes. And we will be the first one basically running these studies, yeah. So the other thing what we did is that we said, well, maybe we could, instead of alleviating carb counting, simplify carb counting. So currently people need to enter the amount of carbohydrate into the app at each meal time. We said, well, maybe we can simplify it. So people, instead of entering the amount of carb, they said, well, the meal is small carb, medium carb, high carb, or very high carb. So this might sound like a small difference, but for a lot of patients, and instead of trying to figure out whether the meal is 50 or 55, it's easier for them to say, well, this is a medium carb or high carb. So let's simplify it. So we built a newer system. And again, we had to do all the testing, documentation, get health kind of approval with a newer algorithm. And then we're on a randomized control trial that compare full carbohydrate counting with what we call a qualitative meal size estimation. And the question we wanted to answer, is the qualitative meal size estimation non-inferior to carbohydrate counting without giving people any other hormone? So this is only insulin. So we did study where we compared the two in a crossover design, 3 weeks per intervention, 2 sites, so both of them here in Montreal, McGill, and the RCM, and 30 others. Now, what we found is that when people had full carbohydrate counting, the proportion of time spent in target range on average was 74%. However, with the newer system, again, we degraded a bit to around 71%. Now, people say 5% is a clinically significant difference. So anything less than 5%, people don't consider clinically significant, but we still had meaningful reduction. However, we didn't do formal interviews, but when we talked to people informally, most patients were overwhelmingly more supportive for the qualitative meal size estimation. And this is where currently companies are trying to go to. So instead of doing full carb counting, the next step for them is to do meal size categorization. And there is already only one company now in the market that did this, actually approved in the US, submitted by NXT. Now, the last thing we're doing, so we built the newer system, and then we're doing a study with a faster insulin. So we said, well, maybe you don't have to add a pramlintide or empagliflozin. Let's get insulin that is faster. So Lonja is a newer insulin developed by Eli Lilly, which is faster than all current insulins. And we're starting a study where in 12 adults, they do 3 weeks per intervention where we compare carb counting, qualitative meal size categorization, and fully closed loop using a newer system that we built with a Swiss pump that's called Ipsomed. And again, we did all of that engineering at McGill, and we're planning to do the clinical trial. Actually, we just got approval, so we're starting soon. So you can see with the study that we're trying to combine newer drugs with algorithms to either improve glucose control or to alleviate carb counting. And we try to assess these devices using randomized controlled trials, so proper clinical testing. Now, the other group of patients that I mentioned earlier are those who are using multiple daily injections. So people who use multiple daily injections, we can't have an artificial pancreas systems. However, what we could do, we could build medical devices, a mobile app that every week look at the previous week data and tell the patient, well, next week, instead of giving yourself after breakfast 5 units, you should give yourself 7 units. And maybe at night, instead of giving 30 units, you can give yourself 40 units. So we call these decision support systems or open loop algorithms. So again, they're not changing insulin every 10 minutes, but more on a weekly basis. So we built that app, and that app is a class 2 medical device. So we got Health Canada approval, and we started running multiple studies with this for this particular group of patients. So just to give you some high level, so if at any minute every week you have the glucose level of the patient, the insulin injection, the amount of carbohydrate. So let's say this is the real data from one of our patients. If you look at night, sugar levels are high. After every meal, sugar levels are low. So can you adjust these insulin dosing parameters? So the basal dose, the carbohydrate ratio, which is basically how much insulin they should take for every 10 grams of carb. Or if people are taking fixed meal doses, how much they should be taking. And what we call correction factor. If your sugar levels are high, how much insulin should you be getting? So these parameters currently the patient uses to give themselves insulin. Can we actually optimize these based on the sensor readings and based on the information of the meal and the insulin delivery using advanced algorithms? So we developed the algorithms, and the first thing we did is a study at a diabetes camp. So this is a camp dedicated for people with type 1 diabetes, where we compared changes every morning done by a physician to the insulin doses for 11 kids versus changes done automatically by the algorithms for 10 kids. And we compared the two outcomes in 11 days. This is actually a pilot study, so we did not have statistical power to actually look at significant p-values and proper hypothesis testing and so on. Now, the first thing you can see here, and this is actually a group of patients that were using our systems. And what you can see here is that both the algorithm and the physician, so the lower row is the physician, the upper row is the algorithm, they actually, each color represents a different patient, and the physician and the algorithm made adjustment over time. So you can see, for example, this particular patient, the algorithm brought down the basal dose from 100% of what they were getting before the camp into 60% by day 7. This patient needed a bit more insulin, and so on. And the other thing, what we found is that for both the algorithm and the physician, the amount of changes or the magnitude of change also decrease over time. So again, this was a pilot study to give us a feeling about how the algorithm works. Now, also what we did, because this was the first study with this algorithm, every morning before we implement the algorithm change, we had the physician looking at it. We had 6 different physicians, and they approved almost 92% of the algorithm recommendation. And there were only 2.4% what we call complete disagreement. So the physician wanted to increase the insulin, and the algorithm wanted to decrease the insulin. And we had similar glycemic outcomes between the two. So then we built the app. So it's an app which is a class 2 medical device, and that basically at mealtime, people choose if they're eating breakfast, lunch, dinner, or bedtime. They enter their sugar level, the amount of carb, and they give a recommendation. And that app basically over time can individualize the insulin doses for each patient. And we build the platform, so the app will send the data online, and then we can see us or the physician, the sugar level of the patient every week, the amount of insulin they're getting between weeks to week, and the sugar level on a daily basis. We can zoom in and look at what happened at every single meal. So we build that app on this is actually a web platform that we can see the sugar level of the patient. And then we said, okay, let's go and test this app using a randomized control trial. So again, you can see similar with the closed loop studies. We build the algorithm, we do basic pilot trials, and then we build the proper software-based medical devices, and then we move into randomized control trial. So we just finished the recruitment of 84 adults with their A1C above 7.5. So these people have suboptimal control, where we compared what we call sensor-augmented multiple daily injections with the app. So people had a sensor, had the app that does the calculation for them, versus sensor-augmented the injection, plus the app, plus a learning algorithm that actually adjusted the doses of the patient every week automatically. And the primary outcome is their A1C. So we wanted to see how much the A1C is improved if we have a learning algorithm compared to the app itself. So the study is not, we finished recruitment, but in actually a month, we will finish the results of the last patient. But this sample patient where the app increased the basal dose from 20 units to 25 every week, and decrease the carb ratio, so the amount of insulin they take at mealtime. And you can see that the time in range was actually went up from week to week. You can see there is some randomness, but on average, it's actually going up if you look at the trend. Another patient, the app actually reduced insulin dose from 80 units to around 40 or 50 units. But at the same time, interestingly, it actually increased the insulin dose. But you can see here the proportion of time these people spending a low sugar level is actually dropping. So this patient had a lot of hypoglycemia early on, but the app increased the basal to actually improve the sugar level. So this shows you that, again, these are sample patients. We will look at the population outcomes after the study, but there are some individuals where you can see clearly the algorithm is improving their glucose control by making automatic adjustment to their insulin doses. Now, physicians can do this, but the patient see the physician once every 3 months or 6 months. And however, with the app, we can make these changes one every week. Now, again, we did interview with the patient. So this patient said, referring to the algorithm, I was actually surprised. The fast-acting recommendations, this patient meant the recommendation at mealtime, were pretty much on par with what I anticipated. But what I was surprised with was the long-acting insulin recommendation, which is the insulin at night. We dropped the basal significantly, which has improved, eliminated my concerns for having low sugars overnight, and has still maintained a steady level throughout my actual waking time of the day. Now, this patient commented the fact that they see their physician every 6 months. So 6-month visit to the hospital, to let that go for such a long period of time could have negative consequences to one's health. This at least gives, a guidance immediately in real time. So the patient man the app. And again, it does give the patient the option to override. So we don't always force this insulin on the patient. We give them the option to override. Another patient said, changing the doses is something we rely on medical professionals to do. I've worked on changing them by myself at times, and there always that fear, have I gone too far? So if the application is as safe as professional, then it is like having that contact in a regular patient. Now, this patient actually was on the control arm, so just commented on the app. I like the fact that it kept track of how much insulin I had in my body at the time, so it won't overdose me, which I cannot do on my own. Because currently, if you give yourself insulin, it works for 5 hours. So if you try to give yourself insulin in 2 hours, you forget how much you gave and how much of that insulin you previously gave still working. So that is all done by the app. So it's really nice because we're getting quantitative feedback when we look at the glycemic outcome, but also qualitative feedback by doing these interviews. Now, one thing, and I will finish maybe in 5 or 10 minutes, but one thing we said, well, let's compare the algorithm to a group of endocrinologists. So let's forget about glycemic outcomes. Let's see how the algorithm compare if you give them certain amount of data and you give a physicians the same data and you tell them make adjustments. So we are currently running a study in 16 endocrinologists where we compare them to the algorithm. So we give them data for 10 patients and they look at the data and the physician said, well, I would increase basal, I would decrease the breakfast ratio and so on. And we compared that to the algorithm recommendation. And they did that for 10 individuals weekly. They repeated that, the same data, also weekly for 4 weeks, and they did looking at a bi-weekly data. So when you looked at the data, okay, someone asking about exercise, this is coming. Now, what we found is that what we call full agreement between physicians is actually only 50%. So only 50% of the time, the physicians among themselves, this showed you the subjective nature of these changes, fully agreed with themselves. But also, if you look at the algorithm versus the physician, we were also around 50%. Partial agreement means the physician, one physician wants to increase the basal, the other one says, well, I don't want to change it, also around 40 or 50%. Now, full disagreement was around 5% among the physicians, but also between the physicians and the algorithm. So that shows you, again, we still haven't finished part 2 and part 3, but this preliminary data is showing us that the algorithm can be seen as a particular physician because it has an average similar approval rate to other physicians and similar disagreement to other physicians. Now, exercise. I received a question about exercise. So one thing we said, well, when patient ate a meal, we updated the app and we said, is this a high-fat meal? Because if you had fat meal, you need actually more insulin later. And if you're doing exercise, you need less insulin at meal time. So we made them announce the exercise. And we developed an algorithm based on reinforcement learning that actually learn for each individual how much insulin they should take for a particular exercise, and we make adjusting accordingly. So quickly data, what we found is that after exercise, the proportion of time people spend in hypoglycemia, it was a single arm trial, so it's not a randomized control trial. That dropped from 5% a day, which is around an hour and 15 minutes per day, to only 15 minutes per day. So people when they do exercise after meal, if you look at the 5-hour post meal, they had actually 20% less, well, 80% less low sugar level compared to the baseline period. So now we need to confirm this in randomized control trial. Now, interestingly, if you look at the patient, you can see there is large variability between patients. So on average, if someone is doing exercise after the meal, we reduce the amount of insulin by 30%. Some patients needed 50% because they do cardio. Some patients needed 20% because they see walking. So we saw a lot of variability between patients, between meals, which really highlighted the need of personalized algorithms. Now, just an idea about innovation. So these companies all developed what we call smart insulin pens. These all came in the last 10 years, which is basically a pen that tracks the insulin and communicate with the phone, or a cap that actually you can put it on a pen and track your insulin doses. So these are all hardware developments that allow the implementation of decision support systems. And also there are multiple companies that came to the market that actually just came because just all recent companies that also allow you to fully close the system. So the development of glucose sensor, it's really opened this whole field of diabetes technology where we start to see innovation on the pen side and on the insulin pump side to allow the development of these smart connected cares, basically. Now, we worked with Bigfoot. So Bigfoot is one of the companies who built the cap. They actually acquired one of our algorithms that make adjustments for people who are using a multiple daily injection. So we work as well with companies, yeah. Now, another thing, there are multiple companies who build apps that actually can communicate with insulin pumps. So Camp, the app, is actually in Europe. And what I heard is that the pump company was selling a good amount of insulin pump before actually working with this app. But then since they started working with this app, they actually sales increased by tenfold because people suddenly get better glucose outcomes. And also start to see improvement on the drug side. So companies start to develop either faster insulin or basically co-formation of insulin pamlintide or maybe glucagon to be used on a pump. So again, all these innovations are basically there driven by the initial trigger of continuous glucose sensors. Now, this class AP is actually our app that we developed here at McGill. Initially, it was acquired by Eli Lilly, which were planning to develop a patch pump, and then they worked with a Swiss company to build an insulin pump. And then here is McGill University. So we were public working with them, and they had a program for 7 years. And then just last year, they decided to shut down the program, not because they had any negative data, but because they had a lot of success with a drug which is more powerful than Ozempic called tirzepatide or Mounjaro that reduces body weight on average by 20%. So they felt, and they have apparently other drugs coming to the market that were very successful. So they decided to focus on drugs that are done to do closed loop devices. Yeah. And we did with them multiple studies. Now, the last slide I want to show is that there's a lot of enthusiasm from the patient side for this therapy. So the group of patients, so if you do hashtag we're not waiting, that actually patients, family of patients, friend of patients that went to hack insulin pumps and the glucose sensors and build their own closed loop systems before any commercial closed loop system became available. So around 5 years before commercial closed loop system became available, patients were already building these systems, hacking them. And then that movement that started an open source community, so you can go to GitHub, download actually the code. It ended up being a company called Tidepool, and Tidepool received FDA approval to be commercially available. So again, a community-led innovation that started by patient hacking insulin pump and glucose sensor. After a year, people were happy. They wrapped it into a company, and that company actually got approval. And again, this is a purely innovation led by patient, family of patient, and basically friend of patients. Now, the other thing, what we do ourselves, we do community engagement. So there's a camp called Camp Coronis. So if you have type 1 diabetes and you are 12, 13 years old, it's very hard for a camp to accept you because they don't know how much insulin to give you. They don't know how to change your infusion set and so on. So what we see basically in major cities, there are camps dedicated for diabetes. So there's a camp here in Santa Gat, which is around an hour away from Montreal, that started by a pediatrician at McGill, I think 62 years ago. And every year they have 250 kids. They spend there 2 weeks. Now, these are the tents of the camp. We do studies there. Now, this is the map. So each white dot here is actually a tent. And this is the boy side. Actually, this is the boy side here, and this is the girl side. Now, when we were doing our studies, we had to spend around 90 minutes to go over all these tents. And, you know, the kids at night, they have low sugar level, high sugar level, and you don't know about them until the next morning if they're wearing glucose sensors. So what we did, we actually built for them a system that does remote monitoring for the entire camp area. So we use a device called Meow Meow. We put that on a commercial Abbott glucose sensor. Then we used the software that was developed by the We Are Not Waiting community. So we took that from GitHub, we made some modification. We covered the entire tent area by Wi-Fi, and we put a phone under each bed of each kid. So we could get the data from Meow Meow to the sensor, to the phone, using the Night Scout, and then we send the data to the infirmary, which is actually here in the middle. And then we donated for them big screen, and the nurses were able to follow the sugar level of each patient in real time with all kinds of alarms. So before the patient have hypoglycemia 4 in the morning, they know sugar level is having hypo, they will walk all the way to the tent, they wake up the kid, and they give them sugar before they drop to low sugar level. So we never published this. Actually, it was done by students who were doing capstone here at McGill, and I joke, maybe it's not a joke, I tell people this is the most important thing we did because, you know, we do our research, we publish. If we don't publish, someone else will do the work. But this camp, if we didn't do that work for them, no one would have done it. So this was a really nice practical application for the diabetes technology field. Yeah. The other thing, we do Cafe Scientific. So we did that twice. Every 3 years, we invite 100 patients with their families, and we talk about research. So this poster, we had someone in real time as we present. She was actually drawing, and we generated this figure by the end of the Cafe Scientific. And I think I'm over time. Thank you. This is my last slide.
Thank you very much, Dr. Haidar, for that wonderful presentation. Are there any questions? Please feel free to unmute, or you can type in the chat. So are there any current developments in, because as you are talking, I was thinking about since we are living in the era of AI, are there any developments that might lead to self-learning insulin dosing algorithms using AI?
Yeah, I mean, so the pump currently most people use techniques from control engineering. Now in the open loop side, what we have done is reinforcement learning, which is technically it's a machine learning approach. But if you mean like deep learning with these multi-layer neural networks and they need a lot of data, and they're just like our problem is not. You don't need complex models to solve our problems. You need a lot of what we call knowledge in the field itself. So sometimes you see people, they work on a computer and they develop this complex algorithm, but they don't really work because they don't fully understand the chemical problem. So yeah, so there are more and more work from AI. What we have seen now with mostly traditional machine learning approaches, so reinforcement learning approaches rather than deep learning approaches. Yeah.
Okay. What about non-invasive monitoring methods that you put on the skin and you don't have to insert?
Yeah, so that one, so there are people who try to develop a glucose sensor using infrared or microwave, and this one I joke with people that I see them at a conference. Next year I don't see them. I see different companies. So for some reason it's been quite challenging. So I think what's happening is that they struggle with accuracy so far. But we see every 2-3 years we see 2 small startups and then they disappear after a while. So I think, I mean, the advantage of this one will become cheaper, and they're not. I mean, these glucose sensors are not invasive. So the current glucose, I mean, they're minimal invasive. But when they came to the market, within 2 years, their use in kids between 2 to 6 years old was increased by 10-fold.
Okay.
So people use it, even very young kids, and people you wear it, it's there for 14 days. But they're quite expensive. So each sensor costs for 2 weeks around $100. And you can do the math. Now in Quebec, they're covered for adults and pediatrics, but a lot of countries they're not covered. In Quebec, they cover pumps for pediatrics, not for adults, but the glucose sensor are developed for everyone. I think in Ontario, pumps are covered, but not a glucose sensor, but they're expensive. So if you have non-invasive one, will be cheaper and nicer, definitely. Yeah, but we don't have them yet. Yeah.
Okay. Any other questions? Just unmute and ask. Mike?
Sure. Yes, Dr. Haidar, thanks again for accepting our invitation to present very impressive data. It's quite, I was very much impressed with your system, the closed-loop system that you have in the algorithm. So I had a question actually related to if you have like infections, let's say you're sick, some viral infection or whatnot. I mean, glucose levels spike, I guess, during infections. Can your algorithm sense these changes?
Yeah, so when people are sick, people need more insulin and their sugar level increase. So initially, the first commercial closed system, the recommendation was if you are sick, then you need to switch off the closed system and manage your sugar level on your own. And the reason is that we all have all kinds of safety rules in the system. We don't go and allow the system to deliver 5 times more than an individual need because it might be sensitive and accurate. So for safety, we have all kinds of rules. But someone is sick, they indeed needs a lot of insulin. So the current systems, I believe still all of them have that caveat. If you're sick, you need to manage your sugar level on your own. I believe so. I don't know the most recent system, but definitely the earlier ones, this was the recommendation, yeah. And again, because we can develop closed system that give you a lot of insulin, but then you have to remove all the safety rules, and then you might have a hypo somewhere else. And what I tell people, developing Bluetooth system is harder than developing an autopilot for an airplane because you need to have one algorithm that works for all patients. And each patient, even within the same patient, you have large variability from day to day. Well, if you have an airplane, you have the use state feedback controller with again scheduling. You change, you test, you change, you test, you change, you test. It works. That's it. You're good for the next 20 years. You don't have that variability that we see in medical applications. Yeah.
My other question was, there's more of a general question for these devices in particular. Nothing specific, but let's say, if we're using phones, for instance, for the app, something happens to your phone, sometimes they're unreliable, maybe you have an issue, it crashes or so on. Is there like a fail-safe in these devices?
Yeah, absolutely. Yeah, so as part of our health kind of mission, as part of the risk analysis, people might forget their phone, the battery might die. So the way we do it, it's more technical detail, but the way we do it, the pump actually is programmed all the time to give you the patient usual insulin, and the phone override that insulin temporarily for the next 20 minutes based on the closed loop recommendation. So what happens if the phone dies after 20 minutes, you fall back to your current to your open loop therapy. Otherwise, if you change it, and then let's say you change it and you give 5 times more than they need, but then the phone dies, they're getting that amount. So it's more technical detail, yeah. And I had a question in the chat about type 2. So the reason people started with type 1, because type 2 diabetes, if you measure sugar level 4 times a day, you connect them, you pretty much have an idea about the sugar level of the individual. Not all of them, because some type 2 look like really type 1, but a lot of them, the sugar level, we didn't feel that the need to measure sugar level continuously. While type 1, the variation in sugar level are really high. However, the glucose sensors now are being used more and more in type 2 diabetes, even in people who do not actually take insulin. And more companies are doing studies in type 2 diabetes. So there have been published closed loop studies in type 2 diabetes. So we're moving slowly toward type 2. Now, type 1 diabetes is around 10% or even 5% of the entire diabetes population. So if you only use these technologies in 10% of the type 2, you actually double your market share. And this is why all these companies are pushing towards type 2, and they're hoping to get only 5 or 10% of that population. So it's a huge, I mean, in Canada, we have 300,000 type 1. In Quebec, I think 70,000. Type 2, you're talking about 2 million, you know? It's a much bigger market. But then if you want to get coverage for them, that's a different story. I was talking to one of the pump companies. They try to, talk to, regulatory agencies and the reimbursement bodies to cover it. And they said, we never mention type 2. Because if you mention type 2 and they start plugging the numbers, it becomes too expensive for them. Yeah.
Okay, well, thank you so much.
My pleasure, yeah. And type 2, they're mostly, I mean, in the past, they used to say half of them, they need insulin at some point. Now we have drugs for type 2 that are quite powerful, that less and less people need insulin. We still don't understand because these drugs are new. So one of them is the SGLT2 inhibitor that I just mentioned, and Ozempic. So Ozempic basically, or semaglutide, just you add these two before people go into insulin. So we don't know yet in the long term how many of them they would need insulin. But still, again, without insulin, a lot of people still use these devices because they need to measure sugar level in a continuous manner. It is very convenient and so on.
I guess the long-term effects of these drugs is not currently known. So we'll find out later the issues with them in these patients.
You mean safety? No, they did. I mean, they did 5 years, 6 years. I think one study went to 7 years or 6 years. So we have done relatively long-term trial, and people continue to look at their adverse event longer. So I didn't mean in terms of safety, but I meant in terms of how many people. Like previously, we know that 50% of people with type 2 on average need insulin at some point. We don't know now. I don't have the numbers 10% because of these new insulin, new non-insulin drugs. Maybe a lot of them. I mean, there was a debate recently in a conference where someone was saying we don't need insulin anymore, and someone was saying you're crazy, this is science fiction, we still need insulin. So again, within the community, there is still genuine uncertainty about how many people still need insulin. So, but still, there's still room for continuous glucose monitoring in the entire type 2 population. But for pumps and injections, maybe less and less.
Okay.
So I would imagine the list pro insulin, the one that you said.
Lunjab, yeah.
Yes. So I would imagine that it works for a shorter duration. So
Exactly.
So do you have to inject more often than?
No. So currently, rapid acting insulins, what we had in the past is Lispro and Aspart, so Humalog and Navorapid, and these ones they work within 30 minutes, they peak around an hour, and they work for 5 hours. Now, Novo Nordisk developed Fiasp, which is a bit faster than Aspart, and Lilly developed Lunjab, which is a bit faster than Lispro. Now, in a pump, you actually don't need to change because the pump gives you a micro bolus every 3 minutes. But what will happen is that at mealtime, previously the sugar level, there's a lot of actually, there is some mismatch between the sugar level absorption and the insulin absorption. I'll show you here in the figure. I think this is a good. Yeah, so you can see here, for example, at lunch or dinner, sugar level goes up and then it drops down. And what happened is that the food absorption is much faster than the insulin absorption. So sugar level goes up and then insulin starts to work and then sugar level drop down. So when you have a faster insulin, this peak will actually be lower because you have a little bit better match between the glucose absorption from the meal and the insulin absorption from the insulin. So you don't need to inject more often, and the idea is that it gives you better control. So I don't have this answer the question.
Yeah.
Yeah, and we're using it for fully closed loop because again, in this case, the patient eats 96 grams, sugar level goes up, and you start giving this insulin, and you really want faster insulin that start to catch up quickly with the sugar level. So what we know, sugar level after, depending on the type of the meal. So if you eat mashed potato, different than macaroni and cheese, but like sometimes after half an hour or 1 hour, only 25% of the sugar in the meal was observed. So you don't need insulin to work very quickly to catch the remaining 75% that's still in the gut, basically.
Thank you.
Yeah. And pramlintide delay the food absorption. So it's always, you either try to delay the food absorption or try basically to accelerate insulin. Yeah.
Any other questions?
Each insulin, you need slightly different algorithm. So the faster the insulin is, the more aggressive you can be on the insulin. Yeah. So you need to change a little bit in the algorithm. You still keep the same architecture, but you change some of the parameter setting. Yeah.
So when you're doing your clinical trials, do you not include those people who have erratic eating?
Yeah, so we have usually a bunch of inclusion-exclusion criteria. So we try, so in your run clinical study, you want to keep it open so that you have generalizability. You cannot recruit everyone the same. But also, if you have a small study of 30 people and you keep it completely open, you start to get outliers that actually changes your outcome. So in our studies, on average, we try to exclude people with high A1C, so people with extremely suboptimal glucose control, people with gastroparesis. So if someone have a condition that actually is slow absorption, we try not to give them a study with pramlintide. People who had severe hypoglycemia in the last 3 months. So we have some exclusion criteria, but we try not to keep it a lot of them because otherwise it can impact the generalizability of our outcomes. Yeah.
Yeah, because I was looking at your other data there, which is really nice, very wonderful, and they seem to have their lunch, and and then there's a peak, and then it goes down, because some people don't have those kind of.
So these are sample patients. So these are the best patients. This is why I like to show them to explain the concept, but then I always show population level. I didn't show any p-values today. I thought we don't have time, but I usually, even when I have only one topic, I show p-values to make sure we show people the entire picture. And some engineers, I'm an engineer, so I don't mean to be like bash all engineers, but some engineers, the first time they come to the field, they do one patient and they go around, they say they cure diabetes because they don't understand. They have to do, there's the reason you do randomized control trials, it's proper control arm. So we're trying to be rigorous when it comes to both the engineering sciences and the clinical sciences. So all our studies are randomized control trials, and if they're not, we try to make sure we don't overstate our findings. So we remind people it's a pilot study. We have to confirm it in randomized control trials. Yeah.
Very good. Very good. Yeah. N01 usually you hear, yes, these engineers, but yeah, very smart. Yeah.
So students, no question. I see 70 people here.
Yeah, students.
And the question came in the chat came from only one person. So maybe the mistake was letting them have their camera off because I'm not sure how many of them were actually listening. Any question? So in my class when I teach, I don't allow cameras off. My rule is camera always on. Any question?
Muskan?
Yes.
Any questions?
No, actually, I was looking at the data, and actually, I love the system. My dad usually uses this one, and I think it will be a great one in future. I don't have any question because yesterday only my dad was like telling me about all this, and today I came to know more in detail about it. Thank you.
Type 1 or type 2, if I may ask?
Type 1.
Type 1, okay.
Yes.
So, what about nutritional supplements and herbal medicines? Do you, some people use them as adjunct kind of?
Yeah, I don't know. So one clinician, I asked I mean, most of them, there's no evidence to support them. So, you know, it's like my dad has type 2, and every time I see him, he tells me, well, I heard okra brings sugar level down, or onion or parsley. So what I do with them, I tell them, well, you can try your okra, but make sure you take your medication. So I think a lot of these
Right.
I mean, unless there is a randomized control trial, there is no evidence for any of that. And when I talk to, I mean, I'm not clinician, so I don't see patients, but when I talk to one of my clinician colleagues, he told me the same. He told me like he hears this all the time, and you don't argue. He says, okay, you can try. I mean, it doesn't harm to eat a lot of okra. Just make sure you take your medication. Yeah, but not to my knowledge, none of the official guidelines support, you know, some of that. Yeah, I mean, of course, there are general nutrition guidelines, like healthy eating and stuff like that. But to say you have type 2, don't take metformin and instead take parsley, it just doesn't work. Yeah. Or honey.
Cinnamon apparently helps. No, doesn't cinnamon?
Cinnamon, yeah, I don't know. I mean, someone needs to do a randomized trial. If someone else believes in cinnamon, they should do a randomized control trial. Otherwise, it just doesn't work.
Yeah. Well, thank you very much, Professor Haidar, Ahmad. It's been a pleasure, and thank you for. It's nice to see this is a clearly unmet need, unmet clinical need that is a wonderful, and it helps a lot of patients. And thank you very much for sharing your knowledge. Really appreciate it.
Okay.
Yeah. Thank you, everyone.
Thanks.
Thank you.
Yeah. And bye, everyone.